Hi everyone, my name is Anush Swaminathan. I study biology and philosophy, and my project is about how animal use in non-medical or basic research saves human lives. It's an ethical justification, hopefully, if it persuades you. That's the same thing again. Okay, so I'm gonna start off with a quote. So in 2005, which is the last kind of rough estimate we have about this, 115.3 million animals were used in experiments. This does not include invertebrates, like fruit flies or any of those. These are talking about vertebrate animals. And that's still probably an underestimate. And every week, I kill 30 to 40, right? That's more important for me in terms of my contributions. So what justifies this particular task? Before that, I get to some core definitions. Basic research is research to just increase knowledge, is to increase awareness around you, knowledge of the world, of how things function. Medical research is research that has distinct medical healthcare-related outcomes. So the question that comes up is, why should I be able to use animals for research that seeks to increase knowledge? It has no justifiable or direct, I'll try to argue for that, it has justifiable, but no direct medical outcomes. Just one caveat though. So my argument is catered against people who argue that in cases where animal research would benefit human lives, it is justifiable. There are some people in a minority who argue that no matter what the scenario is, you should not use animals, whether it's to save human lives or cosmetics or anything. My argument is people who concede that much. So the project is to justify the use of animals in basic research, and the idea is to appeal to basic research as a system rather than as an individual experiment one at a time. Ray and John Greek, which are kind of two individuals that run a popular lobby, try to argue that any one experiment, basic research, only less than 1% have direct medical outcomes. And their argument is, well, why should we be funding animal use in those particular types of research? And my argument is, let's appeal to basic research as a system and not as a one-off experiment scheme. So I'm going to start off with a case study. Let's jump into an example first so I can illustrate my point a little better. So von Mering and Minkowski were two scientists in the 1880s who kind of came together and decided that they wanted to figure out what is the driver behind diabetes. They wanted to figure out, really, what does the pancreas do? That was a question they were asking. Diabetes had been characterized for the past 2,000 years. No one knew what organ was driving it, what were the secretions that were combating it. But they started with a simple question of what the pancreas does. They came together, they went up to a lab, and they depancreatized a dog. So they removed the pancreas from a dog. The dog died within 3 days due to incredibly high levels of glucose in the blood, right? And they repeat this 3 times, and they found the same thing. And that's where this first model was born. The pancreas in the dog as the mammalian regulator of glucose homeostasis. Homeostasis being just equilibrium, right? The driver of glucose levels in your body. And then the important thing is that this experiment that showed the pancreas regulated glucose became kind of the framework with which all future experiments in the field were carried out by, right? So this is kind of like a framework model. In 1901, Eugene Opie at Johns Hopkins found out the specific type of cells that have certain secretions in the pancreas. Couldn't characterize the secretions, but there are these different types of cells, and one of them produces secretions. In 1908, you basically have Ernest Scott, who finds that the best way to extract these particular cells is to use some kind of alcohol combination. You have Zwelser in 1912. This is one of the first attempts to actually find a therapeutic use of these secretions. What happens if I just take this crude secretion and inject it in someone? What do I see? I see a drop in glucose levels. And then in 1921, this is Paulescu, who was a Hungarian scientist, and I think had a rightful claim to the Nobel Prize that the inventors of insulin found. But he was actually the one who kind of found a protein that was very similar to the insulin that's described by the next slide people that I'll bring up. And he found that it actually decreases glucose levels in diabetic patients specifically. And then here we have it. In 1922, Banting and Co., Toronto doctors, isolated insulin. They tested it against diabetes, and they found that it brought down sugar levels in diabetic patients. All of these experiments used animals. All of these worked under the framework of that first model. So what do we take away from this? We take away that all these experiments were conducted under the framework of this first model. They took it as an assumption. The first model asked the why, and then these subsequent experiments asked, the first experiment asked the what, and then the other experiments started to ask the how and the why. There was no single basic research experiment that had direct medical effects, right? But as a system, it was moving towards medical breakthroughs. So this kind of comes up with a mechanism of how research transitioned from basic into medical, and how there's a causal link between the two. there's a first animal model as a descriptive model. That was the dog pancreas as a model for glucose regulator in the body. Then you have enhancement of that descriptive model. That addresses the how. How does the pancreas regulate glucose in the body? And then you have a medical breakthrough, kind of like a ripe model. Think of it as a fruit. When it's ripe, it falls off the tree, giving you a medical breakthrough or benefit. And what's really interesting is that those first two parts of this mechanism are distinctly under the domain of basic research. Medical research doesn't touch those questions. It doesn't posit a what, and it doesn't try to address a how and a why. It just takes those two elements and it tries to make it medically applicable. Using that mechanism, I want to analyze another case study in a way where it's not yet reached medical breakthroughs. It's on the way. It's partially there. So I work with circadian clocks. That's why I have to kill 40 mice a week. And this first model that we can look at for circadian clocks was in 1984 by the people who won the Nobel Prize this last year, Hall, Ross, and Young. And it's called the transcriptional translational feedback loop. A lot of terms, let me break it down. You have something in the nucleus, a protein, that makes more proteins, okay? These proteins accumulate outside the nucleus, and when they accumulate to a certain extent, they come back and stop the proteins that were making them to begin with. And this is a daily cycle that spans across 24 hours, and it just keeps working in a loop. When they posited this model, that's pretty much all they knew. They knew analogs in other animals, but they knew that it kind of works in a loop. But now we know these are the proteins involved. This singular proteins don't matter, but you can see that the elucidation of those vague descriptions has been a lot in the past 30 years. But you see all those gray spaces and those green spaces. We still don't know what those are. We know that they exist based on the mass of the complexes that we've measured, but we don't know what those are. So we're still in the enhancement phase. We're figuring out how this first model can be blown up, how it can be elucidated, how the mechanisms of it can be elucidated. But there's still a lot that we don't know. And the reason why it's still in the enhancement phase, before I actually show you the mechanism, I was at a conference last year and I came across a company that was actually trying to take circadian clocks, what we know of it so far, and find medical breakthroughs. Biotech startups at times start to play fast and loose with rules and controls and isolating variables because they're worried about getting seed money and blowing up their results. The company went bankrupt after amassing around $300 million of investment and 5 years of work. So that's probably my example of how it's not ready for medical breakthroughs yet. So the first model is a descriptive model that's been posited. That's the TTFL model from 1984. The enhancement of the model is taking place within the mammalian system. So you need animals for the first model. You need animals for the enhancement of the model. And both of those are under basic research. And it still hasn't gone to the medical breakthrough yet. And that's what we're waiting on. So as a conclusion, medical breakthroughs need basic research. Simply because it's not just a one-off scheme. It's not just that basic research is a data mine for some medical research programs. It so happens that if you remove basic research, you take the legs of the stool of which humanity sits in terms of medical breakthroughs. So we need animal use in basic research. That was also a really weird analogy. I just thought of it on the spot, and I'm thinking back, and I probably shouldn't have said that. Anyways, so we need animal use in basic research, and that's basically the general conclusion that I've come to. Thank you. Questions? Yes?
What about basic research that we know is not going to lead to medical breakthroughs? Is animal use still justified in those contexts?
Sure. So that's part of the point I'm trying to make. To find any kind of systems that then lead to medical breakthroughs is only a retrospective enterprise, right? I can look back and I can identify that there's a first model. I can identify that there's this enhancement of the model and there's a breakthrough. But epistemically, I can't point out which program right now will actually add to that knowledge base which can give me medical breakthroughs. So making that value judgment is really, really almost impossible. And that's kind of why you have to go with basic research as a whole, because you never really know which component's useful down the line. Any other questions? Yes?
It's just like a devil's advocate sort of thing.
Sure.
You're talking about like ethics with animal usage and just like killing 40 every week.
Mm-hmm.
How do you justify the fact that you're using much telling the animals that we are a superior species.
So superior in what sense?
In the sense that you have the right to kill 40 a week to do research to protect the human race.
So exactly. So that was the caveat that I began with. I'm not. There are individuals who think that no matter what, we should not use animals, right? Human beings' lives are not worth it. Mice can save cancer patients, not worth it. It just so happens that they're in the minority opinion, and most people who argue against kind of the use of animals argue from utilitarian grounds. So they try to argue that it just so happens that it's not effective. It's not worth your time to use animals to save human lives. So that's kind of what my argument is catered against. In terms of what your question is and what I would personally think about it, so I'm kind of grounded more in the utilitarian sense. So for me, I don't really have problems with using animals to save human lives. There are certain, of course, like other final details, like moral points. Well, why can't you use other individuals or human beings for that matter, right? But that's a different question, and it goes into like moral philosophy. Yeah. Yes.
What about like breakthroughs that don't rely on animal research in the basic research phase? Like, do you have any like literature study of like what percentage of medical breakthroughs relied on basic research that didn't have animals versus that did?
Yeah, so that's a fantastic question, right? Because that's one of the ethical questions that comes up. Well, if I can, now they're having stem cells, you can grow organs on a plate. If I can grow organs on a plate, why do I need animals, right? The question that comes up is where ethics always changes based on the finding of the day, right? Scientifically. But right now, animals are the most predictive model. So if you're looking at organoids, they can't measure systems, for example, right? It's just one organ. You don't know the effect on the liver if you're looking at the heart. So there are a lot of flaws and gene expression ratio problems that happen with organoids. So right now, animal models are still the most predictive. They're not perfect. By any means, they're not perfect. But they're still the most predictive of those lot. But the ethics would change if we get to the point where they are as predictive. Yeah, absolutely. Yes?
My concern has to do with the health and well-being while they're alive. Like as a meat eater, I try to eat low meat because I'm against factory farming. I want my animals to live a life that was similar to what evolution anticipated for them before I kill them for food.
Yeah.
And I can see the same logic applying to this use. How can we make sure, especially with work, primate work, where we have such intelligent animals, how can we make sure that they're living an adequate life before they're killed?
Sure. So adequacy, there's a couple of kind of components. So in terms of like experiential adequacy, animals generally, for the most part, in captivity for research don't have the same experiences as well. So they're not put in the wild. They don't have the same kind of socialization opportunities as animals in the wild. But from just a living kind of food and care and medical care, most lab animals are actually taken care of very well on front of food, because there are some very stringent rules in place from multiple different organizations that come together, and the protocols that they push that animals have to be treated very humanely, they have to be sacrificed humanely, they have to be well fed, they have to stay warm. Torture of animals is not really something that happens in medical research. But the question still remains about experiential. That's a different, because we can't offer the same experiences that they would get in the wild, otherwise it wouldn't be in captivity. So that's kind of one side of life that we can't.
We have to try to make the captivity as humane as possible, like let them live with conspecifics and let them have some semblance of some activity, natural activity.
Sure, and I think that still happens. So they do live with conspecifics. They are interacting with their own group a lot of times, but it's still forced interactions. So there's always going to be some loss, but then they don't have to work for food. I'm not trying to put myself in the mind of an animal in the lab, but I'm just saying that there are some elements that we can't capture, no matter how hard we try. Yes?
How do you combat against researchers becoming kind of blasé about using animals? Because I know experientially that people will start using ethical methods to take care of and to kill lab animals, but then over time, gassing mice takes too long. It's easier just to cut off their heads.
Yeah.
How do you combat against that?
Sure. So the modalities of how many animals and how animals are used that's a really good question. That would kind of be the follow-up, that would be the future direction of this, right? Because I've tried to justify that animals can be used. But that doesn't mean I can use 1 million animals to figure out what protein in my eye does, looks for the color red. We know that, but I'm just saying, right? So the point is, when you're looking at modalities, I tried to give a presentation that wasn't grounded in a particular moral philosophy, whether it's grounded in like some kind of like animals of inherent worth, but they lack this worth, or utilitarian grounds. I tried to kind of separate the two. But if you had to go into those kind of considerations, you would have to take those into account. Yeah. Okay, thank you very much.